All right. It is 7:49, so we are officially back from our intermission. I hope you guys grabbed a beverage, grabbed some food, told your family to shut up and stop walking around naked in the background. Again, send those directly to my email. But let's move right on with our next speaker of the night. So we already heard about some interesting synergies and partnerships that might be pretty applicable to this current quarantine we're in, and now we're going to see how you can overcome certain problems that some people, especially researchers, might face uh, from this particular quarantine. So your next speaker for tonight is Dr. Jeremy Long. He's a marine biologist that has never extracted a golf ball from a whale's blowhole. Serenity now. Because hip hop influenced much of his research, he wants to acknowledge sections of his papers include shout outs to Ice Cube, Snoop Dogg, and Del the Funky Homo Sapien. Well, let's learn from Dr. Jeremy on his talk, How to Homebrew a Marine Lab During a Lockdown. It is now my honor to introduce Dr. Jeremy Long, aka Dr. J-Lo.
What's up, everyone? Quick cheers to anyone that's drinking right now. Let me try to share my screen and get this party started, or keep it going, I should say. Okay, hopefully everything will work here. Can I see a thumbs up that you see my opening screen, How to Homebrew a Marine Lab? Yeah, okay, cool. So the question that I'm going to address today is basically, how do you keep a science experiment going during a pandemic? I'm sure this is a pressing question that you've all wanted to know the answer to. So let's get this show on the road. I see some familiar faces in the audience, but for those of you who don't know me, if you Googled Jeremy Long before you came, you might have found out this guy. He's a porn star or an ex-porn star. That's not me. It's not that kind of show. So my apologies. If you want to sign off now, that's totally cool. I'm down there at the bottom, hit number 4. I am a marine biologist, not a porn star, but I do like sea stars. So anyway, that's the segue here. One of the things that I want to tell you about are some of our recent experiments looking at something called rack. So what you need to know about rack is basically this is seaweed that's been torn off the seafloor. It gets pushed up onto the beaches. The birds that are on the beach, they like to feed on the little animals that live inside the rack. And the seaweed, when it breaks down and decomposes, it leaves a bunch of nutrients on the beach as well. So it's a kind of important thing. And the animals that live on the beach like rack a lot. And one of the types of rack that a lot of the animals really like are called black abalone. This is a picture, a couple of pictures of them. Whenever these animals get a hold of kelp, they love it, they tear it to hell, and they chew it all up. Just as a side note, these animals are endangered animals. They used to be much more common in Southern California, but because humans did a really good job at over-harvesting them, and because of a disease, their numbers have declined dramatically. But there are some places where they seem to be doing okay. So one of the sort of big questions we're interested in understanding in my lab is how changes in the seaweed community might affect these animals that live on the shore, the animals that are interacting with the rack. So in the upper left of this slide, you'll see a picture of native kelp. But what we're increasingly seeing is a change, so that instead of just seeing this native kelp washing up on the shore, we're seeing more and more of this invasive devilweed. This is an invasive seaweed that's from Japan. And we're just really interested in trying to know what effect this invasion is going to have on the animals that interact with seaweed rack. So the mystery that we're sort of set out to explore with the experiments that I'll be telling you about today is trying to understand how these seaweed changes will impact these animals that you see here. So just briefly, the animals are hermit crabs, line shore crabs, abalone, and turban snails. I wanted to give a shout out to my student, Rick DeSantiago, who also is known as the Hood Scientist. He is in the house, but he's also not only an amazing scientist, but a really phenomenal artist. So he did all of these drawings. And Zed, you might consider trying to get him to a future one of these events. So Rick is the dude in the middle. The other person that I need you to meet is Wendy White, who's my super technician. There's no way that I would have even have been in the predicament, great situation of making a decision about moving an experiment into my garage had it not been due to the work of these two individuals. So props and cheers to them. Okay, so the story that I want to tell to you today started back on March 6th of 2020, when a bunch of students basically set up this experiment where we wanted to look at how these different animals would perform on these different seaweeds. So in the upper right corner, you'll see some of the red abalone that we got from our colleagues in La Jolla, and the lower right is the turban snail. And the basic idea here is we're going to feed them one of 4 different treatments. We're going to give them kelp, we're going to give them the invasive seaweed, a mixed diet, or a starvation control. And then we're going to measure how these animals perform on them. And my initial segue here was that I was going to tell you about having done, 15 people participated in this project, and we put in over 1,000 hours of work. But Jessica, like, you totally trumped me on that one. I wrote down 80,000 hours, and then I had to cross it out, and so you had 200,000 hours. So you've got me beat by far. If I had those number of hours, who knows what I would have done should this happen again. But I'm telling you that because I need you to appreciate that we had a lot invested. It wasn't 200,000 hours, but it was 1,000. It was pretty good. So we weren't going to just sort of let it go to the wayside. I just want to share a video of this animal. I know it's probably going to be choppy, but this is one of the red abalone. And one of the things these animals are really good at is flipping over. So if you put them on their back, they can basically stick their foot down and flip themselves completely over. I'm super not flexible, so that's not something that I could ever do, but these invertebrates do it with ease on the reg. And you can imagine that's kind of a good thing because they're super vulnerable when they're upside down like that. Okay, so a week after we started this experiment, the virus, coronavirus, was making its way and increasing, and the alarm bells were going off. And so I panicked. And this is an actual image of my dry erase board. I think it still looks like this because I needed to sort of make a plan of what to do if, for example, CMIL, which is the name of our marine lab, would it be open, yes or no? And if it were to not be open, then I needed to come up with a plan to try to keep the experiment alive. So I won't pat myself on the back much, except in this case, a little bit of planning I think did go a long way. Right around this time, I also came into my garage, and I'm in my garage if you couldn't already tell that now. This is where the experiment was live when it was live. And I brought the tank into it, started to fill it with water, get the seawater to the right conditions, just in case anything else happened. Ruining the plot here. Other things did happen. 2 weeks later, the beaches and parks were closed. That's important for us because we needed to keep on going to the beaches and parks to collect our seaweeds, the parks obviously that are adjacent to the ocean. The other thing that happened is that Governor Newsom basically signed the stay-at-home order, and I needed to get to the beaches and parks, and also I needed to get to the Marine Lab to run the other half of this experiment. I sort of took this situation and took things into my own hands. This is a meme that Rick shared, which I just love, maybe because it gives me props, and sometimes it's good to get props from your students. So I brought this experiment to my garage. As you can see on the right, the responses on Rick's Instagram were quite positive. I like Kelly's comment there. I feel like this year is going to produce some wild publications. I think 100% that's going to be true. So in a couple of years, if you read through the methods sections of papers, I think you will see a lot of creativity, maybe in particular from ecologists. So like Jay-Z, I had 99 problems. I was trying to create a marine lab in my garage in a place that this wasn't plumbed to do that. I could tell you a million stories just on this slide, but the one that I do want to share is that these animals, these red abalone, have a particular ocean pH that they like to live in. And when I got all these animals here in the middle of the night, it was like Thursday, March 19th, when I was sort of frantically bringing this experiment into my garage and these animals, I checked the pH and it was too high. And I'm like, oh, fuck, like this sucks, like all this effort, and then all these animals are gonna die, right? So luckily I have a jacuzzi. And so if you have a jacuzzi, you've got a pH adjuster. And in this case, I use this pH reducer to lower the pH.
No, he's.
And just a reminder to mute yourself because I'll get easily distracted. The pH, this reducer, I just added the teeniest amount and the whole tank just dropped dramatically in pH. And so then I'm like, oh shit, like what am I gonna do now? Well, it turns out we do have pH increasers in our kitchen. So if you add baking soda, you can increase it. And after some fiddling, I got the pH in sort of the right zone that I wanted to be working with. And I was able to get the experiment started in my garage. This science is not pretty science. So I'm highlighting that. Duct tape, salt everywhere, cinder blocks, PVC. I will say that maybe as an ecologist, I came sort of pre-adapted for this because these are some of the tools of our trade. And I would say that maybe we wear this with a badge of pride, that we're able to make experiments happen even in the harshest of circumstances and situations. I don't think the reporter is here that interviewed me, but when he was interviewing me, he said, oh man, like when I imagined a science lab, even in a garage, like I did not imagine cinder blocks and duct tape and baking soda. So I think I took a bit of offense to that and maybe went on the defensive, but I don't think any of that made it into the interview. The next obstacle that I had to overcome was to get that essential worker label. This is a pretty boring part of the story, so I'll just say lots of emails to a lot of people. I acted quickly, so I think that that helped. But before I move on from this slide, I definitely want to make it super clear. I do not put myself in the same category as these other people. Delivery workers, postal workers, grocery workers, healthcare providers. I'll raise a toast to them. And I'll just say that I was thankful that I was able to continue working during this time. Okay, so the third obstacle that I wanted to bring up is that we still needed to get our seaweeds to keep the experiment going. So I woke up on March 27th and I turned on Peaky Blinders because we're in a time where people are sort of sleeping in maybe, but I wasn't. I had to have this on mute, so I had the closed captions on. And in this one episode of this show, the character says, Tommy, I'm 45 years old today, and that's how old I was on that day. So I don't believe in sort of signs and premonitions, but that one's maybe hard to ignore. If you've ever watched that show, I had no idea if this was going to be a good sign or a bad sign, but we'll sort of see based on the rest of the story. So I came to the Mission Bay channel. I hope that you can see my cursor. I made it big, but maybe you can see it. Over here in this area of the beach is where we would collect our kelp usually. And over here at the tip of Mission Point Park is where we would collect the invasive seaweeds. So here's the story of that day with some Lego animation. The first person I encountered was a meter maid in a police car. I told him who I was. He looked me up and down and kind of said like, you know, do you have any proof that you should be here? And I'm like, well, I have this piece of paper that says I'm really important, but I don't know how I got past him, but somehow I made it past the meter maid. I then drove into the parking lot over here and grabbed my buckets and I'm like, oh, stoked, like I'm good to go. We're gonna get the seaweed. So I sort of walked out to the beach and I'm almost there. And there's this like beautiful fresh pile of kelp rack and it looks really healthy. And I'm just about to grab it. And over the speaker system, the people in the lifeguard tower are like, sir, please step away from the seaweed. Retreat to your vehicle. The beaches are closed. And so I'm like, oh, like, are you kidding me? I was so close. And I don't know that this is sort of normally in my character, but instead of going to my vehicle, I headed straight for the lifeguard tower. Maybe tried to act like a boss and let them know what I was doing. And I somehow persuaded them that I should be able to collect. So I collected the kelp, was feeling really good about myself, went back to the car. Because I was gonna have to snorkel to collect the invasive seaweed, I started to put on my wetsuit. And just as I got it fully on, the cops appeared now on motorcycles, and they were just shaking their head and they were riled up, and told me that I shouldn't be doing anything here. And I told them who I was. And I think that at that point they had sort of talked to each other. So they're like, oh, you're the biologist, like, tell us more. So I shared the story. Then I went out and collected the invasive seaweed, again over here at Mission Point Park. I've got both the seaweed species that I need. I'm about to keep it going. I can't wait to text the team and let them know. And then there's a squad of police officers on foot. And so it's like, oh, dude, are you kidding me? And I probably could have approached them and asked for their permission as well. That's not the strategy that I took in this case. They went right, I went left. I sort of ducked behind a bush, made it into my car, and I drove away unscathed with the seaweed that I wanted. So that was a win. Tupac came on the radio. I flashed the West Side sign. Those are all pictures from that day. It was a good day. In the right, you can see the fresh seaweed basically being ready to be added to the abalone containers in the garage. And so the sort of punchline here is that for about a month, I kept this experiment going in my garage. Older scientists like myself often talk about how much they miss doing actual science. So I got my dose of it, but it kicked my ass. So after a month and sort of 6 weeks into the experiment, we called it. I told you a bunch of stories today. So what I'm not going to do, unfortunately, is tell you about a lot of the data. But just to highlight some of the results in text here, for the red abalone, we got a surprising result, which is we saw similar growth rates on kelp, the devilweed, and a mixture of those two diets. That was unexpected based on our previous studies. And then for the turban snail, we actually only got high growth for this species when it was fed the invasive devilweed, and we have no idea why that is. There are a lot of follow-up questions to be asked, certainly. But I do want to leave you with a couple of slides just quickly. The first is that I've been contacted by reporters previously, and I usually wasn't really stoked about talking to them, but I thought I'd give this one a shot. There are 3 things that I learned that maybe will help some of you in the future. The first thing, take out your goddamn earpods, your earbuds, your AirPods, whatever they called. I kept mine in the whole time. And so in the whole interview, you see me with these stupid white things in my ears. So that's pro tip number 1. Pro tip number 2, practice how you introduce yourself. This was an actual quote on the recording. I don't think it made it in the segment, but I said, my first name is Jeremy. My last name is Long. Hi, I'm Jeremy Long. I don't know what human ever has introduced themselves that way. And I will say that my 2 girls were listening to this, and afterwards they're like, what the hell? They didn't say that, but that's what I imagine they said. They're like, why did you introduce yourself that way? And I had no smart answer. Fortunately, that didn't make it into the segment. The last thing that I wanted to share is that I didn't know that there was a soundtrack of science. And as someone who likes hip hop, I thought, okay, well, maybe if there is a soundtrack of science, it might have sort of a dope beat. So let's see if I can do this. Yeah. The first beat is, the first soundtrack is hopefully going to be what it sounded like in my mind. So fingers crossed that this plays for y'all. And it didn't.
But when the coronavirus hit, a lot of those studies were put on hold, except for one man who decided to bring the ocean home with him.
So that's what I imagine. I can't see any of you, so if it's just me talking, I am sorry. Here's the actual clip. That's the actual soundtrack of science, heard first here on CBS 8.
But when the coronavirus hit, a lot of those studies were put on hold, except for one man who decided to bring the ocean home with him.
Snail 80, 9.38. So disappointing. Also, I hope that worked. No idea. So I just want to leave you with this slide here. If you ever want to do what I did, I've got some take-homes for you. So big picture, this was maybe what you should walk away with from my talk. Number one, be prepared. Have cinder blocks, duct tape, baking soda. You're going to need all of those. You're going to need that essential label. You're going to need to be polite to any authorities that may get in the way of you collecting your organisms. It never hurts to have a really good squad. And I don't really know how I'm able to do this, but I work with some amazing students, and it's only because of their hard work that it was even an option to keep this experiment going. And finally, practice how you introduce yourself. Don't introduce yourself in the weird way that I did with the interviewer. And maybe also don't do it as how I did at the beginning of this talk, how I define myself as not a porn star. So with that, thank you for your time. I'm happy to entertain any questions. Peace.
This was excellent. Thank you. Somebody, Alyssa says, yai abs, meaning probably the black abalone, that as resilient as you doing the science.
Yeah.
I think the world is grateful for resilient researchers like you, Jeremy. One question. So how does the invasive species of kelp end up in our beaches or bays?
So I guess it's at least a two-part answer to that question. The first is how does it get here in the first place? And then once it gets here, sort of how does it move around? I guess to answer the first question, the most likely scenario for these organisms is that it's most likely coming over in ballast water. So when you've got shipping tanks that are crossing large swaths of the ocean over short time periods, and then potentially going from one place where the ocean conditions are similar to what ours are like, that might be a really important avenue. And you know, the stories that they tell, and I haven't looked into this recently, so I'm not sure about the current status, but there are some efforts to try to get ships to change their water out at sea to try to prevent that from being a problem. But if you're a ship and you're out at sea and there are big waves, becoming super buoyant suddenly by changing your water is not a super popular thing to do. Once the seaweed gets around, it sort of gets around like anything else, right? So it can hitchhike on other organisms. It can move around via ocean currents. Certainly these rafts, so right, when it detaches and floats to the surface, it can then go somewhere else. So yeah, that's how it gets around.
Does the surprising results show that they've adapted to devilweed and mixed environment compared to previous collections, or has this not been measured in this way previously?
I would say it's probably too early to tell for sure, but this invasive seaweed has only been here for like 15 to 20 years. So for the organisms to respond that quickly over such a short time scale, as far as forming an adaptation, I think that that's probably unlikely. I will say that sometimes we can see these sorts of responses happening faster than you might normally predict. So it's not necessarily something that needs to happen over thousands to tens of thousands of years. So it's an interesting question. I think it's probably unlikely, but we certainly are still really early on to answer that question well.
Great, thank you, Jeremy, so much. We don't have more time for questions, but as I said before, we will channel any other questions that there are to you directly by email or on our Facebook page. And I think that's it for now. I think over to Robert. Thank you, Jeremy, so much.